Protoplast fusion. Introduction. Protoplast fusion is a genetic engineering technique used in plant biotechnology that involves the fusion of two protoplasts, plant cells without their cell walls, from different species or strains. This method allows for the combination of genetic material from the fused protoplasts, enabling hybrid plant creation. This process is commonly used to transfer desirable traits like disease resistance, stress tolerance, and improved yield. Materials required: plant cells from the two species or strains to be fused. 2. Enzymes, cellulase and pectinase, to remove the plant cell walls and form protoplasts. Osmotic stabilizers, such as mannitol or sorbitol, to prevent protoplasts from bursting. 4. Fusion agents, polyethylene glycol, PEG, or sodium nitrate, to induce fusion of protoplasts. Electroporation apparatus, optional, used in electrofusion method, culture media, for the regeneration of fused protoplasts into plants. Sterile equipment, petri dishes, micropipettes, beakers, laminar airflow chamber, for maintaining sterile conditions. Procedure: protoplast isolation. Select healthy plant tissues, leaves, root tips, etc. Treat the plant cells with cellulase and pectinase to remove the cell walls and release protoplasts. Centrifuge the mixture to separate the protoplasts from other cell debris. Wash the protoplasts in an osmotic stabilizer, like mannitol, to prevent bursting. Fusion of protoplasts. Mix the isolated protoplasts from the two plants in a suitable fusion medium. Add a fusion agent such as polyethylene glycol, PEG, or use an electric field, electrofusion, to induce fusion. The membranes of the protoplasts come into contact, and the fusion agent helps them merge into a single, hybrid cell. Culture of hybrid cells. Transfer the fused protoplasts to a culture medium that promotes cell division. Under appropriate conditions, the protoplasts regenerate their cell walls and begin to form colonies. The colonies are allowed to grow into cali, indifferentiated plant cells, which can later be induced to form whole plants through shoot and root formation. Regeneration of hybrid plants. The callus is transferred to a plant regeneration medium, where it develops into a full plant. The new hybrid plants are screened for desirable traits such as disease resistance or stress tolerance. Applications: crop improvement. Creation of hybrids with improved traits like disease resistance, drought tolerance, or higher yield. Overcoming sexual barriers. Protoplast fusion allows hybridization between species that cannot reproduce sexually due to incompatibility. Development of novel varieties. Combining different traits from two parent plants to create new varieties with improved features. Somatic hybridization. Used in the production of somatic hybrids for agricultural or research purposes, helping to introduce new genes into existing plant varieties. Disease and pest resistance. Protoplast fusion is employed to transfer genes responsible for pest or disease resistance from one plant to another.